Hello, I will speak today about omics in network medicine. With the aim to speak about the convergence of omics and network medicine, let me first consider where we are today in the disease understanding. We are in search of a deeper understanding of the basis or the causes of each disease. And I wanted to show you today how, in this era of omics, we can improve our understanding, since omics investigation and the big data that derive from this are shedding new light on interaction among molecules that are gene products, such as RNA and proteins. I show you that the data available today from omics analysis are essential for developing the best network-based analysis for each disease and each individual in order to derive the most precise and personalized therapy. As reported in the article by Shork, that is often reported by Professor Lo Scalzo and R. Smith, almost all therapeutic strategies are imprecise. And even if this article was published in 2015, the situation is still unchanged. The blue patients are those that benefit of a drug or a combination of a drug. So, as you can see, only few patients. The majority of the patients indicated in red receive the same therapeutic schedules, but they do not benefit from them. So, till now, we are able to find molecules, yes, to be target for cure patients in a specific disease, but only in imprecise way, or let's say in incomplete way. Today we have identified some of the mechanisms that underline specific disease and have developed drugs based on this mechanism. However, the understanding of the molecules mechanisms that drive a disease need to be expanded in order to properly treat a large number of patients. That is to increase the number of blue patients referred to this article. A fundamental role in understanding the molecular aspect of disease is played indeed by the omics technologies. Omics technologies indicates all tools that are able to go inside the complexity of the disease and to characterize and possibly quantify set of biological molecules in specific samples of interest for a specific disease. And this is indeed a very complex system. So we can distinguish omics in genomics and epigenomics for DNA analysis, in transcriptomics for RNA, and in proteomics and metabolomics for the analysis of proteins. The different platforms for studying omics were developed progressively in the last about 40 years. Following what? The advancement in biomolecular analysis technologies. Here is a summary of the omics technique developed for the analysis of DNA. So I'm referred to genomics that have been come out over years. In 1977, Frederick Sanger developed the DNA sequencing based on the deoxynucleotide Sanger sequence method, enabling accurate reading of genetic sequences. This is the first generation of sequencing tool. In 1980s, the introduction of PCR, polymerase chain reaction, by Curry mules allowed the amplification of specific DNA sequence. It was better for an easier study way. In 1990, 2003, the Human Genome Project was completed, achieving the first sequencing of the human genome using exactly the Sanger method for the sequencing. Mid-2000, the beginning of the generation sequencing enabled rapid, high throughput sequencing of entire genomes. This is the second generation of sequencing, and it allows short read sequencing, typically about 50 to 300 BP. And in the last years, we have the third generation of sequencing, and it overcomes limitation of the second generation by offering long read sequencing up to millions of bases in real time. I'm speaking about DNA, but also RNA can be analyzed. First platform of these categories was Pacific Bioscience, PacBio, and then more recently, the Oxford Nanopore Technologies is coming out. Here is a summary of the omics techniques developed in the years for the analysis of RNA molecules. So in this case, we are speaking about transcriptomics, and we started with as you can see, in the same period, in 1977, the North blot technique was introduced, inspired by the previously Southern blot used for DNA detection. In the 1980s, 1990 become a standard method for the analysis of messenger RNA expression, lives in different tissue and condition. And these technologies decline with the arrival of the array that indeed in 1999 we have the development of these tools. Microarrays are platforms that allow large-scale analysis of gene expression by detecting mRNA. In 1995, indeed, we have the first study using DNA microarray to analyze gene expression profile in bacteria, and was published in this year. In 1993, we have the technique development to analyze gene expression by quantitative PCR. And the quantitative PCR to detect in more precise way DNA levels use two types of probes, or fluorescent probes, CyberGreen, or Tachman probe. In 2005 to 20, we have the introduction of RNA-seq, powered by NGS technology, gradually replace the microRNAs, offering a more detailed and quantitative analysis of RNA, messenger RNA, but also non-coding RNA. More recent series of omics techniques developed for the analysis of protein. Indeed, as you can see, we start this kind of technology 10 years later respect to genomics and transcriptomics. Here is a short summary. In 2000, we have the development of mass spectrometry techniques for large-scale protein analysis. Techniques as multifold and LC-MS allow a detailed characterization of different kinds of proteins. In 2020, the first integrated quantitative proteomic platforms was such as SciLac, stable isotope labeling by amino acid and cell culture, and iTRAC, isobaric tag for relative and absolute quantification, were also introduced. And also, at the end, we have also immunoligation proteomics that is being developed more recently as a high throughput multiplex immunoassay, enabled to simultaneous detection of and quantification of multiple proteins with a single sample. Immunoligation relies on antibodies or aptamer. Aptamer are referred to single-strand DNA or RNA molecules that specifically bind to target proteins and allow researchers to identify the levels of these proteins with high sensitivity and specificity. But how is that? Although since 1980s, we have developed and used, as I've shown you today, various omics platforms that in this way we were able to measure molecules of interest, DNA, RNA, proteins in all kinds of samples. Why we are still not focused properly, or we are and understood the many molecules aspects that characterize each disease? So this is because we have used for the omics analysis the bulk samples. We have molecules in these kind of samples mixed together like smoothies. That is to say that we collect in molecules measurement of each entire samples, including altered cells and cell of the microenvironment, all mixed. This leads to the knowledge of many molecular aspects, yes, but these aspects are without distinguish where, in which cell of the samples we have this molecular alteration. And so we don't know exactly with this kind of omics that we have used so far, where exactly the alteration take place. So to go further inside the complexity of the disease and to overcome the limitation of the omics analysis of the bulk samples, a new technology has been developed in the field of omics in the last 15 years, and it is the single-cell omics, the single-cell sequencing. Indeed, for these new omics technologies, the sample that is analyzed is first, before the beginning of the analysis, dissociated into individual cells, and with single-cell method, we now can analyze molecular information for each cell of each sample. So we can understand which subpopulation are changing and which kind of subpopulation are the component of a sample. Also, as a core concept of system biology, we can understand that the transcriptomics, for example, information of cells, that is which genes are active, are part of a connected manifold. So it is possible to computationally reconstruct the trajectories from healthy to malignant cells and design strategies not just cure the disease, but also to prevent. Therefore, for just over 10 years, we had access to a technology that allows us to acquire, finally, single-cell specific information compared to previous bulk samples omics, which analyze the samples without distinguished single cell. Well, this technology has provided valuable insight, but it also has limitation. And in the next slide, we can see this limitation. We analyzed for 40 years bulk sample with omics, collecting molecules in measurement as a blended of molecules mixed together like a smoothie. While for the last 10 years, we had access to a single cell omics that allow us to get molecular information of each cell in a tissue, in a sample. The problem is that these technologies need to disrupt cells and so need to disrupt the tissue. So we don't know where cell in the space in the disease samples. Indeed, the space is a very important dimension to understand. For instance, if any molecule change in space corresponds to a spatial gradient, or to understand if one cell is communicating with the surrounding ones. This can be inferred from single-cell data, but these reconstructions are often relying on very strong assumptions that do not hold in most types of tissue, such as tumor tissue. For that, the more recent omics technology are spatial technologies. Spatial technologies are more recent and propose what? Allow to retrieve similar information like single cell, but this is done without disrupting tissue. So it is possible to have space, so the exactly position of the cell in the cells in a sample. So we can now analyze the dimension of the space. So we can say that spatial omics are a new kind of microscope that does not only image 1 or 3 colors at once, but allow to detect more than 20,000 genes at once. You can imagine that this generates a lot of data with many open challenges from the point of view of holding, processing, and actual downstream analysis. So here we are now, and not by chance, in the 2020 Nature Methods declare spatial transcriptomics as the innovative technology of the year. We have today several technologies for both single-cell analysis and spatial transcriptomics, which are briefly presenting these slides. There are different in output, in sensitivity. I will not go in detail all of them because of limited time, but I want to say that the spatial transcriptomics are used to different approach, in situ capture or in situ hybridization. As far as in situ capture, it is a method that uses spatial barcodes, arrays, or slides to capture RNA molecules directly from tissue section. After capturing the RNA sequence, allowing researchers to map gene expression back to its original location in the tissue. While in situ hybridization approach use fluorescent or colorimetric probes that bind specifically to target RNA sequence within the tissue, it enables direct visualization of gene expression patterns while preserving spatial context at a single cell molecule resolution. In the field of spatial omics, we had the opportunity to develop the most recent technology published in June 2024, called OpenST, Spatial Transcriptomics, which represents an advancement in this field. This technology enables to capture a high number of genes for cell with single cell analysis, and also, this is a very important point, molecular resolution at subcellular level, which is not available with the other spatial technologies. This allows the identification also of rare cells and lowly expressed molecules within a tissue, and enable the high-definition reconstruction of the tissue architecture. It highlights molecules that segregate closely, suggesting potential active or inactive signaling pathways in a sample. For this reason, this technology generates a large amount of data per cell, providing molecular insight that can define previous undefined aspects of disease in great detail. Last but not least, the cost of the analysis of each sample is significantly lower compared to other omics procedures. OpenACT is also highly flexible technology that generates data that can be easily integrated with data that comes from other omics, including single-cell omics. But this is important since, together with single-cell omics, spatial omics expanded as well in the field of proteins. So we have indeed a platform, very, very recently set for spatial proteomics. In the slide are reported two representative platforms for this that allow a large-scale analysis of protein and their localization and dynamics within a tissue. Imaging-based spatial proteomics methods enable quantitative spatial analysis of over 40 proteins markers across a whole tissue section in a very single cell resolution. So we are now in the multi-omics era with single cell resolution from all kinds of omics, particularly transcriptomics, spatial transcriptomics, single cell transcriptomics, proteomics, spatial transcriptomics. So all within tissue. This is the very last novelty. So omics, particularly spatial omics and single cell omics, generates massive biological data sets that indeed are fundamental for the understanding of biological networks. So I'm referring to gene, protein interaction. And to highlight these, I show you this data on the constant increase in the number of scientific papers that report data resulted from single cell and spatial omics. Based on what have been presented today, I can conclude that spatial omics are adequate approaches to define new disease molecular aspects, to identify target and choose specific disease target for one or combined drugs, and also to identify biomarkers of a specific stage of a disease that can be used for better stratify patient. For a successful outcome of the data collection through such omics technologies, network-based integrated approaches are needed. And here is the convergence of omics and network medicine. And by making this interaction with network medicine approach, we can personalize treatment and improve patient outcome. That is to say, for us, increase the number of blue patients referred to Schwark paper. Despite its promise, omics faces several challenges. Big data complexity. Integrating multi-omics data remains computationally intensive. And all knows this point. And we have also ethical issue. Personalized medicine raises concern about genetic privacy and accessibility. And finally, for the future direction, the next frontiers involve developing more accurate network-based biomarkers and improving tools for personalized medicine. In conclusion, spatial omics are now for sure reshaping our understanding of disease. And network medicine, since is an approach that treats disease as disturbances in interconnected biological system rather than isolated molecular defects, as well described by paper from Professor Lo Scalzo, is the appropriate approach to integrate vast biological data sets that come from omics, and is the strategy to develop precision therapeutic schedules and deliver a better and more personalized healthcare. We, in the last year, apply the network approaches, and we published results obtained in a prospective study enrolling patients with prostate cancer suspicious who underwent MRI-directed fusion biopsy, and circulating microRNA evaluation. We analyzed samples with omics, and in collaboration with Professor Valeria Panebianco and Professor Paola Paci, we made a network-based analysis to identify MRI biomarkers and microRNA drivers of clinical significant cases. With the integration of omics data and network-based approach, we were able to propose an integrated pathways for early detection of prostate cancer that allow accurate patient allocation to biopsy and patient stratification to risk group categories, reducing overdiagnosis and overtreatment of clinically insignificant prostate cancer patients. The time is finished. I end here, and thank you for your attention. And I look forward to participating in the discussion later. Thank you.